Classification of amino acids. There are more than 200 amino acids in nature, but human body contains only about 60 amino acids. But on hydrolysis, proteins yield only 25 amino acids. Out of these, only 11 amino acids are essential amino acids. Out of the 25 amino acids, 23 amino acids are alpha amino acids, and the remaining 2, proline and hydroxyproline, are amino acids. Amino acids containing amino group in the place of amino group. Amino acids are classified in various ways. One is classification based on their incorporation in proteins. Second one is classification based on the structure of the side chain. Number 3, classification based on the position of amino group. Number 4, classification based on reaction in solution. Number 5, classification based on polarity. Number 6, classification based on biological importance. Classification of amino acids based on their incorporation in proteins. At present, more than 200 amino acids have been identified in animals and plants. Still, not all of them serve as constituents of proteins. Thus, based on their incorporation in proteins, amino acids are classified into two groups. They are protogenic amino acids and non-protogenic amino acids. Protogenic amino acids. The amino acids which are used for synthesis of proteins are called protogenic or proteinogenic amino acids. They are usually components of proteins. The protogenic amino acids are of two types, namely major amino acids and rare amino acids. The major amino acids are widely involved in the synthesis of vast number of proteins, irrespective of the species concerned. The 20 amino acids from alanine to valine listed in the table are major amino acids. The rare amino acids are the derivatives of the 20 major amino acids. Example: hydroxyproline, hydroxylysine, aminocitric acid, asparagine and glutamine. Now coming to the non-protogenic amino acids. A majority of amino acids are not incorporated into the proteins. These amino acids are called non-protogenic amino acids. They do not participate in protein synthesis. They occur in the cells either in the free state as metabolites or as part of non-protein compounds. Example: ornithine, citrulline, gamma-aminobutyric acid, beta-alanine, pantothenic acid, thyroxine and many others. Certain non-protogenic amino acids such as canavanine, digicolic acid and beta-cyanoalanine, isolated from plants or toxins for man. Classification based on the structure of side chain. On the basis of the structure of the side chain R group, that is, amino acids are classified into three types. One is aliphatic amino acids, second one is aromatic amino acids, third one is heterocyclic amino acids. First one is aliphatic amino acids. The amino acids containing straight chain in the R group are called aliphatic amino acids. The aliphatic amino acids are further classified into four types based on the number of amino and carboxylic groups in the side chain. Number one is monoamino monocarboxylic acids. Second one is monoamino dicarboxylic acids. Third one is diamino monocarboxylic acids. Fourth one is diamino dicarboxylic acids. Now, monoamino monocarboxylic acids. These amino acids have one amino group and one carboxyl group in the structure. These are neutral amino acids. Example: glycine, alanine, serine, cysteine, threonine, methionine, valine, leucine and isoleucine. Monoamino dicarboxylic acids. These amino acids contain one amino group and two carboxylic groups. These are acidic amino acids. Example: aspartic acid and glutamic acid and the derivatives asparagine and glutamine. Third one is diamino monocarboxylic acid. These amino acids contain two amino group and one carboxylic group. These are also called as basic amino acids. Example, lysine, hydroxylysine and arginine. Next, fourth one is diamino dicarboxylic acids. These amino acids contain 2 amino group and 2 carboxylic group. Example, cysteine and dicysteine. The next one is aromatic amino acids. The aromatic amino acids contain aromatic rings. They are monoamino monocarboxylic acids and are neutral in reaction. Example, phenylalanine and tyrosine. Next one is heterocyclic amino acids. These amino acids contain a heterocyclic ring in the structure. The ring may be an imidazole ring or an indole ring. Example, proline, hydroxyproline, histidine and tryptophan. Classification based on the position of the NH2 group. According to the position of the NH2 group, amino acids are classified into 3 groups, namely alpha amino acids, beta amino acids and gamma amino acids. Alpha amino acids, the carbon atom next to the acid group is called alpha carbon atom. If the amino group is attached to the alpha carbon atom, the amino acid is called alpha amino acid. Example, alanine. Next, beta amino acids, the second carbon atom from the acid group is called beta carbon atom. When the amino acid is attached to the beta carbon atom, the amino acid is said to be beta amino acid. Example, beta amino propanoic acid. Next one is gamma amino acid. The third carbon atom from the acid group is called gamma carbon atom. When amino group is attached to the gamma carbon atom, the amino acid is called gamma amino acid. Example, gamma amino butyric acid. Classification based on reaction in solution. On the basis of the chemical reaction in solution, amino acids are classified into 3 groups, namely neutral amino acids, acidic amino acids and basic amino acids. First one, neutral amino acids. The amino acids which do not contain any amino group of or carboxylic group in the side chain or R group are called neutral amino acids. They contain one carboxyl group and one amino group, and hence they are neutral in character. Example, glycine, alanine, serine, threonine, valine, leucine, isoleucine, phenylalanine, tyrosine, cysteine, cysteine, methionine, thyroxine, tryptophan, proline, hydroxyproline, etc. Second one is acidic amino acids. The amino acids containing additional carboxylic group in the side chain are called acidic amino acids. As they contain an additional carboxylic group, they impart acidic properties. Example, aspartic acid, glutamic acid, aminocitric acid, asparagine, hydroxyglutamic acid and glutamine, etc. The third one is basic amino acids. The amino acids carrying an additional amino group in the side chain are called basic amino acids. They impart basic properties. Example, lysine, arginine, histidine, ornithine, etc. Classification of amino acids on the basis of the polarity of side chain. Amino acids are classified into 2 groups based on their tendency to react with water. They are hydrophobic amino acids and hydrophilic amino acids. The first one is hydrophilic amino acids. In hydrophilic amino acids, the side chains have high affinity for water. Hydrophilic amino acids are either electrically charged or uncharged. The charged side chains are attract water dipoles. The side chains may be positively charged and basic. Example, lysine, arginine, histidine, etc. Negatively charged and acidic amino acids are aspartic acid, glutamic acid. And uncharged hydrophilic side chains can readily participate in hydrogen bond formation with water. Example, glycine, serine, threonine, asparagine, glutamine, cysteine and methionine. Next one is hydrophobic amino acids. In hydrophobic amino acids, the side chains do not interact with water. The hydrophobic amino acids may be aliphatic amino acids. Example, alanine, valine, leucine and isoleucine. or aromatic amino acids, example phenylalanine, tyrosine, tryptophan, etc. or amino acids also, example proline. Classification of amino acids on the basis of biological importance. Based on their inclusion in the diet, amino acids are classified into two groups, namely essential amino acids and non-essential amino acids. Essential amino acids. Certain amino acids cannot be synthesized by organisms. They must be compulsorily included in the diet. They are called as essential amino acids for their normal health. These amino acids are called essential amino acids. For human being, about 11 amino acids are essential. The examples are arginine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, valine and tyrosine. Non-essential amino acids. Certain amino acids can be synthesized in the cells from essential amino acids or other compounds. Hence, these amino acids need not to be included in the diet. These amino acids are called non-essential amino acids. The amino acids other than essential amino acids are called non-essential amino acids. They are alanine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, hydroxyglutamic acid, glycine, hydroxyproline, proline, serine. New amino acids. In addition to 20 L amino acids that take part in protein synthesis, recently two more new amino acids are described. They are, one is selenocysteine, 21st amino acid. Next one is pyrrolysine, 22nd amino acid. Selenocysteine. Selenocysteine is recently introduced as 21st amino acid. Selenocysteine occurs at the active site of several enzymes. Next one is pyrrolysine. Recently it has been claimed as 22nd amino acid by some scientists. The stop codon UAG can code for pyrrolysine. Modified amino acids. Any derivative of an amino acid resulting from reaction at an amino group, carboxyl group, side chain, functional groups or from the replacement of any hydrogen by the heteroatom. The definition normally excludes the peptide containing amino acids. Epinephrine and non-epinephrine and catecholamines. Both are modified amino acids derived from tyrosine. Non-standard amino acids. Besides the 20 standard amino acids described above, present in the protein structure, there are several other amino acids which are biologically important. These include the amino acid derivatives found in proteins, non-protein amino acids performing specialized functions, and the D amino acids. One is amino acid derivatives in proteins. The 20 amino acids can be incorporated into proteins due to the presence of universal genetic code. Some of these amino acids undergo specific mutation after the protein synthesis occurs. These derivatives of amino acids are very important for protein structure and functions. Selected examples are given here. One is collagen, the most abundant protein in mammals, contains 4-hydroxyproline and 5-hydroxylysine. Histones, the proteins found in association with DNA, contain many methylated and phosphorylated or acetylated amino acids. Next one is J-carboxyglutamic acid, found in certain plasma proteins involved in blood clotting. Next one is cysteine, is formed by combination of two cysteine. The cysteine is also considered as derivative of amino acids. Next one is non-protein amino acids. These amino acids, although never found in proteins, perform several biologically important functions. They may be either D or non-D amino acids. A selected list of these amino acids along with their functions is given in the table already. Next, D amino acids. The vast majority of amino acids isolated from animals and plants are L category. Certain D amino acids are also found in the antibiotics, antinomycin D, valinomycin, etc. The D-serine and D-aspartate are found in brain tissue. D-glutamic acid and D-alanine are present in the bacterial cell wall. That amino acids useful as drugs. There are certain non-standard amino acids that are used as drugs. D-penicillamine, D-dimethylglycine, a metabolite of penicillin, is employed in the chelation therapy. Wilson's disease. This is possible since D-phenylenamine can efficiently chelate copper. Next one is N-acetylcysteine, used in cystic fibrosis and chronic renal insufficiency, as it can be functions as antioxidant. Functions of amino acids. In particular, 20 very important amino acids are crucial for life, as they contain peptides and proteins and are known to be the building blocks for all living things. The linear sequence of amino acid residues in the polypeptide chain determines the three-dimensional configuration of your protein, and the structure of protein determines its functions. Amino acids are imperative for sustaining the health of the human body. They largely promote the production of hormones, structure of muscles, human nervous system's healthy functioning, the health of vital organs, normal cellular structure. The amino acids are used by various tissues to synthesize proteins and to produce nitrogen containing compounds, example purine, heme, creatine, epinephrine, or they are oxidized to produce energy. The breakdown of both dietary and tissue proteins yield nitrogen containing substrates and carbon skeletons. The nitrogen containing substrates are used in the biosynthesis of purines, pyrimidines, neurotransmitters, hormones, porphyrins, and non-essential amino acids. The carbon skeletons are used as a fuel source in the citric acid cycle, used for gluconeogenesis or used in fatty acid synthesis.